This is a production of Cornell University.
All right, so today I'm going to be talking about the secret life of beets. How does azoxystrobin impact the beet rhizosphere microbiome? I'm especially interested in how this relates to disease control, particularly Rhizoctonia solani. So Rhizoctonia is the #1 beet root rot pathogen here in New York. Rhizoctonia solani AG group 2.2 causes root rot of table beets, and this fungus is a problem throughout the season. It reduces stand establishment and causes damping off, and also continues to infect developing and mature roots, which decrease final marketable yield as well as limit the prices and the payments made to growers. As a result, azoxystrobin, a QOI fungicide, is applied annually to almost every beet field in the state without regard to actual risk of resistance or off-target effects on the soil health and the microbiome. Now, these off-target effects are what I'm especially interested in right now. So because it's a fungicide, azoxystrobin has a huge effect on the fungal community in the soil and in the beet root microbiome. However, we've seen research has shown that bacteria are also affected, either directly because they eat azoxystrobin or because they are affected through relationships with these impacted fungal populations. And we can actually see that there's a shift in bacterial populations as azoxystrobin levels are increased and applications are made over a longer period of time. This decreasing bacterial diversity, the microbiome diversity in general, is important because it's a well-known fact that diverse microbiomes are associated with low disease levels. In order to manage disease, perhaps we can support microbiome health and minimize other inputs we need to make. Some recent research from Europe has made the connection between sugar beets and the root microbiome, finding distinct differences between healthy and diseased sugar beets. We can see the healthy beets shown here in yellow and green, while the red diseased beets are shown in red. Now, I like to look at this picture like this. It's actually two beets I've photoshopped together, but there's a lot going on, that we can't see when we look at a healthy or diseased beet. All these microbes, they're working together. They have a consortium of different relationships and different species that are promoting root health through a variety of functions, either by direct disease antagonism, pathogen antagonism, or maybe outcompeting space or other resources with the pathogen, or even supporting the plant's defense system. So I want to know, and what I'm going to look at over the next 2 years is how can we support this healthy and diverse microbiome, and will that have an effect on disease levels? I have two questions that I will be looking at. How does azoxystrobin impact the structure and diversity of the rhizosphere microbiome? And two, how does the rhizosphere microbiome and the impact of azoxystrobin affect root disease caused by Rhizoctonia solani? So I planned a series of field trials where the treatments are azoxystrobin or no azoxystrobin and Rhizoctonia inoculum or no inoculum. I'll be applying the inoculum 4 weeks after planting to minimize the residual effects of azoxystrobin. I want to see what happens to that microbiome and then how that microbiome affects disease development. I'll be sampling the microbiome at 2 and 8 weeks post-emergence, extraction, sequencing, and OTU analysis, the whole works. While at the same time, I'll be taking disease ratings at 2-week intervals, starting 2 weeks post-inoculation and culminating in harvest metrics at the end of the season. So by combining this data, I hope to see that what we see or what we can't see within the beet and on the outside of the beet is having an effect on disease development and whether this plays a role in Rhizoctonia solani disease risk. I want to emphasize again how excited I am to be pursuing this research, and I think it could be a real step in sustainable and practical disease management in the future. Thank you for listening.
This has been a production of Cornell University, on the web at cornell.edu.